Welcome back, everyone, to the OIS Retina podcast. This is Firas Rahal. I'm in Los Angeles in my group here, Retina Vitreous Associates. I'm also a partner in New York City-based Excite Ventures. I'm delighted to have as a guest today Dr. Megan Baldwin of Ophthea, a company you're going to hear about today. I'm sure most of the retina practitioners have already heard about Ophthea. They've been in clinical trials for some time. Welcome, Megan. Thank you for joining the program.
Thank you, Firas, and happy to be here.
Save the date as OIS returns to San Diego on December 1st and 2nd. Our 13th flagship summit unites corporate, clinical, and capital leaders driving novel therapies for both the front and back of the eye and focuses on addressing unmet needs across all of ophthalmology and optometry. Head over to OIS.net and click on events for more information. Your path to innovation begins here.
So, Megan, I always start with a little bit of a brief personal history. I find that for me, that's often the most interesting, is learning about where people kind of came into our space from and their background. So I read that your degree, a PhD, was in something referred to as a PhD in medicine from the University of Melbourne. Tell us about that degree. What does a PhD in medicine mean exactly out of that university?
Yeah, so I'm from Melbourne, and I'm coming to you today from Melbourne, living here. Apart from travel, this is where I spend most of my time. So I did my undergraduate degree at the University of Melbourne, and then in Australia you do an honours year after you've done your undergraduate, and then you move into a PhD. And the PhD in medicine was essentially doing a PhD studies. I actually focused on the biology of VEGF-D, which is a new member of the VEGF family that was discovered just prior to my undertaking of my PhD. But I did that through the Faculty of Medicine, based at a hospital that was just adjacent to the University of Melbourne. And I actually was based at the Ludwig Institute for Cancer Research at a time when anti-angiogenic compounds were being developed for the treatment of solid tumours in the cancer field. And it was really only a few years later that the eye disease opportunity came through as well.
So that brought you to your early career, and I have some notes on that. And I knew about some of this because I've been in the space a long time. So early on, you did work in angiogenesis and or treatments related to angiogenesis at Genentech, now Roche, and it looks like you worked with the great Napoleon Ferrara, who's it's been well documented what he achieved there and how some of those developments came about. What do you remember about that time, notably related to ranibizumab and bevacizumab, two drugs that are near and dear to most retina doctors? And what was your experience at that time?
Yes, so I joined Genentech, as I said, to do a postdoc after I'd done my PhD here in Melbourne, and that was really to build on my understanding of the pathway and the biology of the VEGF family members. So I thought it was very much a good fit for my scientific background. I joined Genentech when there was about 3,000 people working at the company. I think by the time I left, a bit over 5 years later, there was close to 14,000, and the timing was quite interesting in the sense that bevacizumab or Avastin at that point didn't have an approval, and in fact it was a time where everyone sort of understood the potential of anti-angiogenic compounds, but they were yet to sort of get through the approval process, and I think there was some very interesting clinical data at that time. But once it got that approval in colorectal cancer at the time, it really took off, and the growth of Genentech at that time was really extraordinary. It was a very exciting time to work at the company. Working in Napoleone Ferrara's group was a really fantastic opportunity for me. Obviously, he has led the way both in the cancer setting, but also in ophthalmology in terms of targeting this pathway. And he's been pivotal in bringing very clinically successful drugs to market. And it was a real honour to be able to work with someone who's been so important in that basic research side, and seeing that come through translational development into blockbuster drugs for the treatment of two very important indications.
Well, just for the record, because we're going to come back to it later, but specifically at this point, since they came up, these drugs we just referred to block which VEGF subtype? Because I know we're going to talk a little bit more in detail later.
Yeah, so they block VEGF-A, so that's the first member of the VEGF family, as you know, and that is the mechanism of essentially all of the standard of care treatments that we have for retinal eye disease now. They all converge and basically do the same. They work in the same way by blocking VEGF-A, which was the first member of the VEGF family to be discovered. But of course, we know now that there are 5 members of the VEGF family. Obviously, Ophthea is very interested in VEGF-C and VEGF-D, which have overlapping receptor binding activities. And by blocking VEGF-C and D, we can actually have an impact on the same receptor pathway that can drive angiogenesis as well as vascular permeability as well.
This is brilliant. I'm definitely coming back to that. But with regard to your original career move over to Ophthea, how did those early experiences with Genentech then lead to your presence at Ophthea or the current development plans at Ophthea? How did you start off there, and what was the original projects you were involved with that then led to what you're doing now?
Yeah, so my background as a PhD researcher was to generate knockout mouse animal models. During my PhD, I'd created the VEGF-D knockout mouse. When I went to Hans-Peter Gerber's and Napoleon Ferrara's group at Genentech, they were looking at a lot of transgenic mouse models where there were models of VEGF-A overexpression, and they were looking at different Avastin molecules and kind of follow-on versions of Avastin, if you will, in those mouse models in order to understand what binding affinity and receptor specificity, etc., did to the biology of the animal. So for me, it was about using animal models to get an insight into drug development and what different approaches might actually have on the outcomes for disease. So it was really it was a basic research study that I was involved in, but it had a direct impact on my interest in drug development. And I think at Genentech, it being a very large and very successful company, and very different to what I'd done during my PhD, I got exposure to a lot of different aspects of drug development, and I was able to move across into the commercial side of the company by working in the market planning department, doing a lot of competitive and intelligent activities that gave me exposure to a lot of different groups, business development, regulatory, clinical, whilst I was working in that commercial side. And having had that broader experience within Genentech meant that I was kind of well placed to come back to Australia and support the former CEO of the company that is now Ophthea. He had at the time brought in intellectual property around the VEGF-C, D, and VEGF receptor 3 pathway. There was various parties that had generated that IP. One of them was the group that I'd worked in for my PhD, and it was a really good fit for me to be able to say, okay, well, I was involved in a lot of that early stage research. I had my name on some of the patents. I was very familiar with the work that had been done, and it was just a great fit at that time for me to move back to Australia, where the idea was to really look at how we can translate that basic research into therapies. And I joined the company in 2007 in order to firstly study oncology, but we pivoted to ophthalmology several years later, and that is what Ophthea is now, an ophthalmology-focused company.
Yeah, I'm glad you brought that up because I didn't know this history either. I'm familiar with the company, obviously, in more recent years as an ophthalmology company. It looks like some trials were initiated in 2015. It was previously a cancer, again, solid tumor cancer therapy company. Has that gone away completely now and it's strictly ophthalmology?
Strictly ophthalmology. Our IP does actually cover not only cancer, but a lot of other potential indications that we may be able to use our intellectual property for. But in reality, all of our resources, all of our focus is on developing OPT302 or sozinibercept. That's the VEGF-CD trap molecule. That is the molecule that is specifically going to be used for ophthalmology. It will never be a drug that we administer systemically for cancer treatment. It is an ophthalmology-focused company. And for many years now, we've been focused on getting our phase 1, phase 2 clinical trials completed. And now we are obviously in very much advanced through our phase 3 clinical development with that trap molecule as well.
That transition to becoming an ophthalmology, a retina, specifically a retina ophthalmology company, what do you recall about that? How was that decision made? Was it just what was happening in the market around the world for these retina therapeutics, or were there other factors involved? You were there during that transition, I'm assuming.
We were there, and it was a really big decision because for the company, as a small biotech company based in Australia, that had been around, frankly, for a long time, there was a lot of expectation that we were going to continue with our oncology program. And at that time, we were developing specific selective antibodies against VEGF-C and against VEGF-D. But the trap molecule was looking very, very promising in animal models of eye disease. We'd taken our antibody through phase 1 clinical development for oncology. The oncology space and the development opportunity, it was very, very competitive. There were a lot of companies around the world looking to develop assets for cancer treatment that were targeting the VEGF pathway. And it was just really a reflection of the fact that I think when you're running a company like this, you've really got to follow the science, you've got to follow the data, you've got to look to see, well, where does our molecule work best and what is the biggest opportunity? And so it was a variety of different reasons, but it really has to come down to the business case. It has to come down to the strength of the data, and you have to let that follow you when you make really big decisions about dropping one area of research and focusing on another. And I think that we absolutely made the right decision. The molecule that we have has obviously a broader profile of activity because it does block VEGF-C and VEGF-D, and we immediately thought that that was a better approach than looking at selective antibodies to one or the other molecule. So it was really the strength of the data, and I think that data has continued to play out in the strength of our clinical trial outcomes as well now.
Yeah, it takes a lot of courage to make those decisions. It seems in retrospect when they work that, wow, well, no brainer. Not at all. I've been involved in such decisions, and it's a really scary time when you're making such a commitment and abandoning a long-held business plan that you're pivoting from. So I congratulate you for that, and it looks like you guys are doing well, so it looks like you made the right decision. Very difficult, though. Prospectively speaking, those are hard things to do. I'm glad you used the term VEGF trap. Those of us who were around during the development of aflibercept remember this term very well. It got coined. Maybe you could explain with regard to VEGF-C and D specifically, or more broadly, it's up to you, to the listeners who may not be familiar with that term, what is meant by trap or VEGF trap? What's the mechanism of action here, specifically of your molecule and or any others that might refer to themselves as a trap?
Yes, well, I think a lot of the listeners will be very aware of monoclonal antibodies, which bind targets, and they do very well at binding and inhibiting those targets from binding receptors. And a trap molecule conceptually does the same thing, but the trap molecule is a different protein structure. It's actually comprised of the endogenous receptor binding domain from VEGF receptors. When we think of aflibercept, as you said, that is also a trap molecule. That is the receptor binding domains from VEGF receptor 1 and VEGF receptor 2. Our molecule is similar broadly in structure, but our binding domain is actually the first 3 extracellular ligand binding domains from VEGF receptor 3. And that is why we have our own unique differentiated intellectual property position around this trap, because it is comprised of VEGF receptor 3's binding domain. And that binding domain very, very tightly binds to, and it's evolved to bind to VEGF-C and D. So you think of that as like a protein domain that acts as the trap, and then on the tail end of that binding domain is human IgG1 Fc, which is basically the constant fragment or the antibody Fc arm that is on human IgG1 immunoglobulin. So it has an antibody arm, but a binding domain that is from an endogenous receptor. And traps now have been in clinical development, not only in ophthalmology, but also in other diseases as well for many, many years. They bind very tightly. They have a really high affinity. They tend to be well tolerated because they are comprised of an endogenous protein. And we've certainly found that our molecule is very well tolerated as well. It's a big molecule. It's 140 kilodaltons. It's even slightly larger than aflibercept because it does have 3 domains. But it's a protein biologic molecule similar to an antibody, but not the same as an antibody.
So in a sense, and this is a simplistic view, instead of blocking the receptor, mimicking the ligand, you're going to mimic the receptor in solution, essentially, which then binds the potential ligand, blocking it from then binding the receptor itself.
Yeah, it's a soluble form of the receptor, as you say, which mimics the receptor, and then it mops up or traps the ligand. And then when it traps the VEGF-C and D, it means that C and D is no longer available to bind to the receptor that's bound to the cell surface. So it actually acts as an inhibitor.
And
exactly
in the current paradigm, the drug that's out there that we're using a lot now that does this similar mechanism is aflibercept and not the others. Is that correct?
That's right. Yeah, bevacizumab is a full-length antibody. We know Lucentis is the binding or the Fab fragment of an antibody. So they are really more monoclonal antibody derived. Aflibercept is the trap molecule, and Eylea OPT302 is a trap as well.
Thank you. That clarifies a lot. I think there is a lot of, I don't know about a lot of confusion, but maybe not so great an understanding in the day-to-day use of these drugs that these two different paradigms exist. Thank you for clarifying that. I want to move a little bit into the clinical part of your program, which is where you are now. Let's go back historically a little bit. You announced positive data in phase 2 trials for both wet AMD, correct me if I'm wrong, and DME in 2019 and 2020 respectively. If so, I'm correct. Can you tell us about that data set or those data sets that came out and how those protocols were done and what you concluded from them?
Yes. So we reported our wet AMD trial results, which was a phase 2B clinical trial in treatment-naive wet AMD patients. We recruited 366 patients into that clinical study, and it was a 3-arm study. We looked at OPT302 combination effect of OPT302 with ranibizumab at 2 doses. So it was a 2 milligram dose of OPT302, which is the dose that we've taken forward into phase 3. And an OPT302 0.5 milligram dose in combination with ranibizumab. And then we compared those 2 dosing arms compared to ranibizumab dosed every 4 weeks according to label. And in that phase 2B clinical trial, we dosed patients every 4 weeks for 6 months, and we read out our data. And we demonstrated superior gain in visual acuity when OPT302 was administered at 2 milligrams on top of ranibizumab compared to ranibizumab alone. So we were getting a superior visual acuity benefit. Across the total patient population, it was 3.4 letters over and above ranibizumab alone. When we dived into that data in more detail, we identified that it was the majority of patients in that clinical trial that had minimally classic and occult lesions. And these are the lesions that we know tend not to respond quite so well to anti-VEGF-A treatment. But that is where we saw the greatest benefit of the VEGF-C and D inhibition with OPT302. And across that group, which was, as I said, the majority of patients that was recruited into that clinical study, over 80%, we saw a 5.7 letter gain over and above standard of care. So we saw an additional and over line gain of visual acuity benefit. The patients that were in that combination treatment group actually got over 16 letters of visual acuity gain compared to baseline. And I think that's quite an extraordinary visual acuity benefit that we really have never seen before. And there's no other company that is currently in clinical development that's demonstrated superior results like that. And I think when we look at some of the recent data, albeit in very successful clinically launched drugs now, where there's between 6 and 7 letters of gain from baseline, and we're showing 16 letters of gain from baseline in our phase 2b. I think you can see that, you know, the real opportunity here is to show additional benefit for patients, actually improve upon clinical efficacy and outcomes. And we may well be able to take our expectation of what is the expectation for visual acuity gain in these patients beyond what we'd ever thought that would be possible before. So we're very excited about that phase 2b data in wet AMD. After we reported out that large clinical trial in the phase 2b setting, we also reported out our diabetic macular edema phase 2a study. This was a smaller study. Obviously, it was a phase 2a. As opposed to the phase 2b. It was powered in a different way, and we recruited actually quite a difficult DME population of patients into that clinical trial. All of the patients that were recruited into that study had been on prior anti-VEGF-A treatment, so they were at various points in their responsiveness to either Avastin, Lucentis, or Eylea. And then what we were seeking to do is to add on OPT-302 into those patients and then look at additional clinical benefit in those patients that hadn't done so well after in many cases, many years of anti-VEGF-A prior treatment. That was a noisier study, it's fair to say, and I think you see that when you do studies that are very heterogeneous in their patient population. But what we did was, when we teased out those patients that had been on Eylea therapy and had good anti-VEGF-A suppression going into our clinical trial, we were able then to see similar visual acuity gains than what we'd seen in the wet AMD study. So we saw advantages of OPT-302 being added on to treatment, resulting in superior clinical benefit, superior vision in those patients that received the combination treatment. So that gave us a second disease indication that has demonstrated the benefit of targeting VEGF-C and D over and above VEGF-A. We are currently really focused on our wet AMD phase 3 clinical studies, but in the long run, we see opportunity to also take this molecule into the DME setting as well. And I think the next step for us is to explore efficacy of OPT-302 in DME patients in a phase 3 setting. But realistically, we would be anticipating doing that after the readout of our wet AMD trial.
Great. And I want to come into the phase 3 trial in a moment and have you speak to us a little about that. But I was so glad that you brought up minimally classic or occult choroidal neovascularization. I certainly was well into practice when we were using those terms a lot before the anti-VEGF era, where everybody just got injected and no one was really caring to look at these subclasses. And most of us who treated in those days with focal laser or PDT and the like did recognize that those morphological differences on angiography or other imaging did relate in a great deal to the outcomes and which treatments may or may not be successful. And I've been sort of surprised that's kind of gone away, because I think it was a very interesting and useful tool. It sounds like there may be some utility in that paradigm, again, perhaps with these different VEGF, C and D blockers, so to speak. Or do you foresee now the coming of an era where we're going to treat more specifically towards certain morphological appearances like we were trying to do back in those days of the pre-VEGF treatments?
I don't think we'll need to have our patients classified per se in the real world, because we are seeing efficacy across the board, and we do see the greatest efficacy in the majority of patients that have those lesions. But I think you're exactly right. The field has moved away from using the classifications, and that's really a reflection of the fact that the anti-VEGF-A or the VEGF-A inhibitors have worked in all of those lesion types. But when it comes to biology, looking at the morphology of the vascular network and the degree of leakiness and fluid, as we move towards more tailored therapies and actually move towards having more therapeutic options available for physicians, I think you're exactly right. It may be that we start thinking about the morphological characteristics of particular patients and their lesion types in order to guide and select for treatment. As I said, I don't think that that's necessarily going to be needed for OPT302 at all, but I think there is a move to want to tailor treatments so that you get the best possible benefit, and giving that choice and putting that kind of knowledge in the hands of the physician so that they can tailor that treatment is how we're going to build on and actually maximize clinical benefit across the board. So I do think that is where the world of science and biology will go. And I think in other disease settings, like in oncology, biomarkers and characterizing lesion types, etc., is all very, very clinically important. And I think we'll see that starting to come into ophthalmology, but it'll take a little bit of time.
Yeah, I agree. And I certainly think these drugs work across the board to an extent, but in the interest of personalizing healthcare, and the people are going to the extent of, genetic personalization, and I'm all for it.
Right.
We have some simple tools at our behest, like fluorescein angiography or maybe other biomarkers on the OCT that undoubtedly have utility. And in the early phases of anti-VEGF therapy, when we first were doing clinical trials with Lucentis, and then ultimately after it was approved, I had the personal observation that the classic lesions did really well really quickly, and no one ever really quantified this, but there may be something there even in the simplistic treatments we're doing now, such that as more drugs come on board, maybe there'll be certain morphologies where we'll know that combination therapy makes a lot of sense even before trying monotherapy. It's just a thought.
Yeah, and I think it makes perfect sense when you think about the classic lesions that are very active lesions, highly angiogenic, very, very leaky, and VEGF-A inhibitors, and VEGF-A itself is a very potent molecule. It actually causes a lot of very acute angiogenesis and a very much a leakage and a permeability type of lesion. So inhibiting anti-VEGF-As, they tend to work very, very well in those lesions, but it speaks to other parts of the pathway actually having a greater role and actually explaining why different lesions look different, and tailoring that and understanding the biology of where different members of the family may have bigger biological roles is kind of where science is going to go, and that will then start to translate into different tailored therapies for the patient as well. And I think another example for our program that has been very insightful for us is the response of a small group of patients in our trials that actually had polypoidal choroidal vasculopathy, or PCV lesions. And once again, it was this group of patients, which I think across the board are recognised as being quite challenging to treat with VEGF-A inhibitors. Our patient groups in our study, the standard of care ranibizumab didn't work so well in the PCV group, but when we added OPT302 on top, we almost doubled the response. So again, it speaks to perhaps VEGF-C and D are really having the most important role where anti-VEGF-A treatments are not working as well, and that's where we can expect the biggest benefit.
You explain it in a way that makes it very understandable. Thank you. That makes a lot of sense. And I agree with you. I want to give you a chance to speak to your phase 3 clinical trial that you alluded to. I know we're involved in your trials, full disclosure, here in our Los Angeles clinic, so I'm familiar with it. Maybe you can tell us a little bit about the trial that's ongoing right now. I'm assuming it's a registration trial, but you can let us know what are the sort of main protocol arms and treatment arms in that trial, how many patients, et cetera.
Yes, so we have 2 phase 3 registrational studies ongoing right now. We really have designed our clinical studies, and they're large clinical studies, in order to support approval really globally. That's why they are really big studies. We're doing one study, which is our SHORE study, with OPT302 in combination with ranibizumab. And our other study, which is being run independently, is OPT302 in combination with aflibercept or Eylea. And we've been very intentional in wanting to do one of our studies with Lucentis and one of our studies with Eylea in order to support the concept broadly that OPT302 and VEGF-C/D inhibition can be added on to any anti-VEGF-A treatment. We really want to position this as a mechanistic addition, as opposed to OPT302 can only be combined with one or the other agent. We really want to be agnostic with respect to OPT302 being able to add on, regardless of what happens in the anti-VEGF space, whether or not it's gene therapies or small molecules or what have you, or biosimilars or anything that's targeting VEGF-A. We believe OPT302 will be additive. So those phase 3 clinical trials are ongoing. We anticipate enrolling about 990 patients into each of the clinical studies. We're very much advanced through our patient recruitment phase. We're over 3/4 of the way recruited, and we expect to be completing patient enrolment into those trials in the first and second quarter calendar year next year. And then it's 12 months then after that last patient is enrolled to our top line data readout, which we anticipate in that first half of 2025. So we're really getting to the end of our phase 3 clinical program. It's a very exciting time for the company, having, now taken this molecule all the way through. And we're literally, 18 months or so away from what we think will be the biggest catalyst for the company. And based on the data that we've demonstrated to date in our phase 2B setting, where we have really been very conscious about not changing too many aspects of the clinical trial in the phase 2B, and then having that kind of then mimicked in our phase 3, and to the extent of doing our statistical analysis of that data in phase 3 based on our learnings from phase 2, we believe that we have a very good opportunity here and a very compelling case that lends to the confidence we have in our phase 3 outcomes.
Yeah, you certainly have every reason to believe and have this confidence. Good for you. 2025 is not that far off, so you're also very far along. How do you, and I know this is supposition at this point, we're presuming approval and a lot of things to happen, but looking down the road then, given what you've said and how it's structured, is this an adjunct therapy with anti-VEGF drugs that we currently use? Can this be potentially a sole therapy? How do you view it as a marketplace addition if and when you reach approval? Yeah,
well, look, we obviously see the clinical benefit of anti-VEGF-A treatments. I think it's important to block anti-VEGF-A, and we're all about improving the clinical efficacy, improving the clinical outcomes for patients. And I think to do that and to maximize visual acuity benefit for patients, it's about having them receive standard of care and then also receiving VEGF-C/D inhibitor on top of that. Ideally, we would see OPT302 used whenever an anti-VEGF-A treatment is used, in order for the physician to choose the best possible treatment to give the best possible outcome for patients. We recognise that in some cases, patients will be cycled through an anti-VEGF-A treatment and then put onto the combination treatment. We've also shown in our phase 1 setting that when we add OPT302 on its own and actually treat as a monotherapy, that we see clinical benefit with OPT302. And that makes sense because in their own right, VEGF-C and D have a very important role in mediating angiogenesis and lesion development. So there is the chance, as a product that's formulated on its own, that a physician may choose to administer on its own. But I think by and large, this will be a therapy that is administered together with standard of care treatment. And that's how we will actually address the suboptimal outcomes for patients and actually improve visual acuity across the board. But we would hope it's used very early in the treatment course for patients in order to maximize the gains and have those gains last for longer and prevent, the clinical and visual decline that tends to happen with patients over time.
Thank you. And I think I might know your anticipated answer to the next question, but you brought up durability. That's certainly a big discussion point at every retina ophthalmology meeting. I'm constantly talking to people. Companies are hungrily approaching, the durability of these great drugs to be longer and longer. Obviously, you have to look at your drug down the road and kind of, as a business manager, anticipate where it fits in the marketplace that will be there by the time you get there. And that's guessing to some extent, but intelligent guessing. How do you view that with regard to the changing marketplace and dynamic with the longer therapies that have been coming about with newer drugs and potentially with carrier molecules and the like that are in development but not yet ready for prime time. How do you view this in conjunction with that?
Yeah, well, the first thing I'd say is I think it's great that we're moving towards that kind of next generation of more durable anti-VEGF-A treatments. But I think the thing to remember there is, as you know, in the real world setting outside of clinical trials where it's very much a fixed dosing regimen, we fully anticipate that OPT302, just like all of the VEGF-A inhibitors that have been approved, that it will be tailored according to a treat and extend or a PRN treatment mechanism. So we can see that there's the potential for OPT302 to be administered perhaps less frequently than every 8 weeks. That's the kind of data and clinical experience we'll get once it's on the market and once we do further clinical studies with the molecule. So I think we can fit into a landscape where combination treatment, similarly to standard of care treatment nowadays, is tailored according to maximising visual acuity benefit for patients over time. It may be that even if there is a very durable anti-VEGF-A treatment that is out there, that OPT302 could then be administered in follow-up check-in visits with the patient as well. So I think all of that kind of treatment modality and the frequency of treatment will play out once we are approved, and it'll be about maximising outcomes. I think the other important thing to think about in terms of durability of treatment, though, is that with OPT302 being an add-on to standard of care, we fully anticipate to have a superior benefit. And that superior benefit may also result in being able to lengthen out the response as well. So a better benefit that lasts longer is absolutely a feasible outcome for adding OPT302 into the treatment paradigm. So we may actually help not only improve efficacy, but to actually extend durability as well. And I think that that's really important. It's not the outcome, the endpoint of our phase 3 studies, but I think in the long run, it's absolutely worth exploring that we have this additional value of the asset to be able to actually have a better response and extend our treatment interval.
Yeah, I agree totally. And I agree with your point that a lot of this stuff has to play out post-commercialization, as did with the other drugs. Those of us who treat lots of patients figure out our own and in aggregate, with groups of doctors, algorithms that seem to work for various subclasses of patients and continue to evolve them. And I like a lot as a clinician that you're speaking a lot and consistently about visual acuity. Although these drugs have been great and people get better, and yes, we want more durability, there's a lot of vision still on the table. There's a whole massive area of improvement with vision, and some of the frustrations with for the patients, by the patients, isn't just about how frequently they have to be treated. We talk a lot about that. It's also the disappointing visual outcomes that have come about. So even if the durability is still in question, if you improve vision, a lot of people will be very happy and very will look forward to this treatment. I think both measuring sticks are excellent.
Yeah, and I think there's a lot of talk about the durability because there's been a number of companies that have failed with their approach to show the visual acuity benefit. But it's a very hard conversation to deny that, clinical outcome and visual benefit and efficacy is really what we're all about. We want to maximize outcome for patients, but there's no doubt the field has become very focused on durability, but I think that that's as much a reaction to the number of companies playing in that space. But it's Ophthea that's got the intellectual property and the product that can actually turn it on its head. And I think that we'll have a very unique and differentiated position in the market that offers benefit. And that's what's really going to be driving use of the product.
Great. You mentioned DME. Is what next for Opthea after this? You mentioned maybe a registration type trial for DME, and separate from that, are there any other pipeline products that you wanted to mention, if any others other than OPT302?
We do have selective antibodies, but we won't be developing them for eye disease because we see that the OPT302 approach is the optimal approach. And so in terms of, the corporate development, it'll be about expanding the clinical opportunity for OPT302. We want to make sure that on the back of positive phase 3 data that we have a very efficient commercialization strategy for the molecule, and we want to roll it out and make it available for patients across all of the jurisdictions around the world. And we'll be focusing on the US and Europe. We would be hoping to enter into partnerships with larger companies that have a footprint in those regions, and it will be about getting the drug to patients and commercializing this in the most effective way. And then we'll turn our attention also to wanting to expand out the opportunity for OPT302, not only just DME, but then there's retinal vein occlusion as well, and all of those indications that were, it's easy to think of now that Lucentis and Eylea have quite a broad number of indications, but at one point they started with just neovascular AMD, and it was only after approval that there was that rush to get out and the opportunity to actually take the molecules into different disease settings, and that's what we want to do with OPT as well.
One final question on the business side, and you mentioned possibly a readout in early 2025 from these dual phase 3 registration trials. If that were the case, when would you be looking at commercialization? What's a realistic time frame for a clinician to have this in their hand? And given that, are you already building towards that commercialization, or is it premature to have that discussion?
Oh, absolutely. I think when you're doing a late stage program like phase 3, you have to think about commercialization and what it looks like. We've done a lot of work for that, mainly focused on the US, but we do have fast track approval from the FDA with this molecule, which may also accelerate those timelines at the back end post data in order to be able to submit our BLA to the FDA and then not long after the MAA to EMA. So we'll be looking to read out second quarter 25 calendar year, and then realistically probably about a 6-month preparation for the BLA. That's including some of the benefits that we'd get from fast track approval, and then looking for the review and approval of the data package, the regulatory submission in 2026. So it's really not too long away, and it's certainly not too far away to top line data for this company.
That's great. Congratulations. What a great development plan. You outlined it beautifully. Dr. Megan Baldwin, thank you. Thanks a million for coming on. I really learned a lot, and we're going to hear more about VEGF-C and D from you and maybe others. And we'll invite you back if you are so inclined after you get further data.
Great. Thanks very much for having me, and it's a pleasure to speak with you. Thanks.
Thank you.